In the last segment of the course, we discussed the sort of variation that's found in human genomes. In this segment, we're going to move on to talk about a particular subset of variants, those found in monogenic disease. As we learned before, the genome of each individual contains more than 3 million variants from the reference human genome. Because these mutations are almost random, some will have a deleterious effect on the function of one or more genes, and so lead directly to disease. Because these diseases are usually caused by variants in only one gene, they're referred to as monogenic disease and exhibit a Mendelian inheritance pattern. The result is 7,000 so far identified monogenic diseases. Each disease is rare, an incident of 1 in 10,000 lives birth is top end, and for many only one or a few cases have ever been reported. Nevertheless, because there are so many of them, the total disease burden is high, with several % of the population affected. For any particular individual, identifying the causative mutation or mutations has the potential to improve treatment or to allow prenatal diagnosis. What sort of mutations are causative of monogenic disease? There may be some ascertainment bias in our current understanding of this, but the pie chart gives an approximate idea. The largest segment, containing just under half of the mutations, is for missense variants, single base changes in the coding region of a gene that change an amino acid. The second largest segment is for small insertions and deletions, less than 20 bases long. There also is a significant presence of single base changes that destroy splice sites or introduce stop codons, so truncating the polypeptide chain. One example of a well-studied monogenic disease is phenylketonuria, PKU. This is a frequency of about 1 in 10,000 in Caucasians, and if untreated can lead to severe mental developmental problems. Over 100 causative mutations have been found in the relevant gene for phenylalanine hydroxylase. As you can see from the figure, missense causative mutations are scattered around the three-dimensional structure of this enzyme, and many act by destabilizing three-dimensional structure, leading either to impaired folding or shorter half-life, so there is an enzyme deficiency. This is a recessive disease in that both copies of the gene must be affected by mutations for disease symptoms to occur. As is the case for many monogenic diseases, symptoms only appear when the total protein activity is greatly reduced, less than 10 or 20% of wild type. PKU is a relatively simple monogenic disease in that symptoms usually point to just one possible gene. Many such diseases are more complicated, both in the difficulty of precisely describing the symptoms and that for a given symptom list there are a number of genes where the causative mutation might lie. The table illustrates this, showing candidate genes for different types of heart and lung disease, with up to 17 genes for each type and some genes occurring for multiple types. There are now many clinical pipelines designed to find causative mutations, and these have reported success rates of between 25 and 40%. There are many reasons for non-success. First, the mutation may lie in a gene not currently known to be associated with the symptoms. New genes are continuously being added to the monogenic disease lists. Second, most pipelines use sequence data for only coding regions of genes. Thus, mutations that affect, say, chromatin structure, or even just gene expression, may often not be covered. Some clinical centers are now experimenting with the benefits of sequencing complete genomes and also collecting data on chromatin structure variations, as well as larger-scale copy number variations. One such study from the Sick Kids Hospital in Toronto has shown that over 30% of cases of severe developmental disease that were unsolved with simpler sequence data could be solved. A third reason for non-discovery of causative mutations is that many candidate mutations have not been seen sufficiently often to establish a disease relationship and must be assessed by computational methods. As we discussed in the earlier segment, some types of mutation, such as those truncating a polypeptide chain or damaging splicing, are regarded as obvious loss of function, LOF variants. Missense mutations remain a problem. As we saw in the earlier segment, current interpretation methods are based on protein sequence alignment properties and are not sufficiently reliable to meet the current US reporting guidelines, which require 95% or higher confidence for a deleterious assignment. What do the nuts and bolts of a clinical pipeline look like? The input is usually a VCF file that we discussed earlier, containing details of each variant and information about reliability. The first stage of the diagnostic pipeline will usually do further quality analysis on the overall data, looking at whether the ratio of transition to transversion-based changes is as expected, whether the frequency of common and rare variants is consistent with other datasets, and whether there is any apparent strand bias, a tendency for a mutation to be only detected on one of the two strands. The second stage examines rare and novel mutations in the genes of interest, consulting databases that collect known causative mutations for monogenic disease, such as HGMD and ClinVar. These databases also contain confidence information on whether the reported mutations are really likely to be deleterious. There are a number of tools available to assist in variant annotation, and ANNOVAR is a popular one. Inheritance pattern is also considered, and knowledge of parents' variants can often be decisive when available. The quality of each individual variant will also be assessed. In addition, the pipeline will probably use one or more of the missense computational methods. Popular ones are Polyphen and SIFT and CAD, although objective testing has shown that these are not necessarily more accurate than others. Even though the computational methods are not highly reliable, they can be very effective in identifying likely causative mutations. The figure shows one example, a missense mutation in a voltage-gated potassium channel produced from the gene KCNB1. Multiple sequence methods predict this to be deleterious. Caution, because of correlation between the methods, uncalibrated consensus actually may be misleading. It's the only mutation that fits the patient's intractable epilepsy symptoms. Other causative mutations in this protein inhibit repetitive neural firing. From its situation in the structure, this one appears to be involved in regulation of channel opening. There are a number of ongoing developments that will extend the value of mutation analysis. Improved missense computational methods will identify the subset of deleterious assignments that do meet clinical criteria of reliability. More systematic use of protein structure will clarify mechanism, as we saw just now. Use of whole genome sequence and genome structure information, as well as improved candidate gene lists, will extend candidate mutation coverage. Development of detailed models for the function of each protein will provide specific information on the role of each amino acid, used to modulate the general computational methods.